Not too long ago, for detailed analysis of brain networks, researchers had to use reduced preparations, acute slices, organotypic cultures, dissociated neurons. More recently, it became possible to study single neurons and even individual synapses in vivo, using 2-photon microscopy and patch clamp recordings in the brain of anesthetized rodents. Today, leading scientists are increasingly realizing that anesthesia is distorting the brain function. This is why the latest trend in neurosciences is to avoid anesthesia and work on the intact brain of awake, freely moving and behaving mice. But here's the catch. How do you immobilize the animal's head and keep the brain steady for microscopic analysis while the animal's body is moving around, showing behavioral responses to sensory stimuli? Researchers have tried it all. They constrained the animal by wrapping it or squeezing it into a narrow tube, placed the animal on a treadmill, on a spinning disc, on a rotating ball. But all these solutions have significant limitations. Some require computer-generated virtual reality to provide the animal with a meaningful sensory input. We have created a real, tangible environment for the test animals. We call it mobile home cage. A mouse walks on a flat floor of an airlifted cage and moves the cage with its paws. The head is firmly immobilized for microscopic imaging or electrophysiological recordings, while the mouse navigates around the cage, touching and whiskering the walls. The mobile home cage fits easily under the majority of microscope frames and only requires a standard laboratory source of pressurized air to operate. Head fixation is stable enough for a variety of 2-photon microscopy and electrophysiological experiments, such as imaging of cortical vasodynamics and blood flow rate, quantifying morphology and turnover of dendritic spines, and monitoring calcium signals associated with spontaneous or evoked action potentials in cortical neurons. Imaging sessions can be repeated several times a day with short intervals of just a couple of hours, which greatly enhances the value of longitudinal experiments, be it pharmacokinetics of fluorescently labeled antibodies or the time course of neuronal recovery after traumatic brain injury or stroke. Mobile home cage also enables single-cell electrophysiological recordings from different brain regions in awake mice. Importantly, imaging and electrophysiology can be directly combined with behavioral readouts, when animals respond with their body movements and locomotion patterns to a variety of sensory inputs, including sound, smell, lick, whisker touch, or visual stimulation. A behaving mouse with firm head fixation for time-lapse microscopy or patch clamp recording.